The first presentation is Karena Gill, and she is a student from the University of Alabama. Her talk is about the fluid inclusion study of the Smackover Formation in the eastern of Gulf Coast and the implication for Dolomitization models.
All right, thank you. Good afternoon, everybody. Today, I'll be presenting on the fluid inclusion study of the Smackover Formation in the Eastern Gulf Coast and looking at the implication for dolomitization models. My name is Karena Gill, and I'm a PhD student at the University of Alabama. The Upper Jurassic Smackover Formation of the Gulf Coast region, shown in Figure 1, has been the subject of many studies since its discovery in 1922. And so research on the Gulf's Jurassic system over the last half a century has largely been driven by the economic importance of the Smackover as a prolific carbonate reservoir and major hydrocarbon source throughout the Gulf Coast region. Mesozoic deposition of the Gulf Coast region followed the breakup of the Pangea supercontinent and rifting and opening of the Gulf of Mexico, which occurred between the Middle Triassic and Late Jurassic. Dolostones in the deeply buried environments commonly possess higher porosity than limestones. And so an understanding of the extent and distribution of diagenetically altered facies, and specifically the dolomitization, is important for estimating and predicting reservoir quality, especially in the deeply buried prospects of the Smackover Formation in the eastern and southern Gulf of Mexico, where dolomitized upper Smackover facies constitute the most productive reservoirs. The Smackover facies in the field shows low porosity and are relatively less dolomitized compared to the upper Smackover greenstones, which have high porosity and are extensively dolomitized. So the important point here is that Smackover dolostones retain porosity to greater depths of burial than limestone, which is important for increasing reservoir depth for the Smackover. The traditional approach to studying ancient dolomites involves petrographic descriptions, cathodoluminescence, trace element geochemistry, as well as carbon, oxygen, and strontium isotope analysis. So these methods have provided important constraints on the origin of Smackover dolomites and attribute dolomitization to a number of different, sometimes conflicting models. But one overarching theme that repeats most often is the marine evaporative model, in which dolomitization is being driven by the movement of hypersaline brines from the overlying Buckner anhydrite member. So here, my approach to this study is first to characterize the distribution of the dolomite facies in Alabama, and I'll do this using selected core and well data. Then I will constrain the dolomitization models by looking at the different dolomite types and their environments, specifically under petrographic analysis. I will also use cathodoluminescence and then further constrain my models using fluid inclusion data. And then lastly, I will test the dolomitizing environments. So my field area is the Appleton field that is located in the Conecuh subbasin within Alabama. And I have selected 7 wells that are shown here and create that cross section that is outlined in red. So for the petrographic study, I deciphered the paragenetic relationships and understood the recrystallization and providing a baseline for understanding the geochemical signatures. So I use standard thin sections as well as cathodoluminescence to do this. So for the results for this section shown here, I have 3 main types of dolomites. So the first one is D1, which I define as the early stage fine grained dolomicrite. And so this represents a shallow burial, low temperature during early diagenesis, environment. So you can see here that we have the preservation of some of the relic structures within there. You have different oolitic grains that are also preserved, and you also have recrystallization within this species. For our second dolomite type, this is D2 that's shown on the left, and then our third dolomite is D3 that is shown on the right. So these just represent our later stage replacement dolomites, with D2 being an earlier stage, and then D3 being the latest stage of recrystallization for the dolomites. So for D2, we have coarse equant sub to euhedral dolomite rhombs, and then as we move into D3, you have clearly euhedral dolomite rhombs with mostly cloudy cores and clear rims. And these are our inclusion-rich dolomites. And the signature of, and they have a signature of influx of basinal brines. So these are enriched. For the dolomite cement, we have a latest stage pore and fracture filling dolomite, and these represent an enriched signature of dolomitizing fluids. And so they replace our coarse sub to anhedral crystals. So to summarize the petrography of the dolomites and their replacement stages, the first one is our early stage fine grain dolomicrite, representing a shallow burial and low temperature diagenesis. And then we have the later stage D2 and D3, which are our replacement dolomites, and they go from coarse, and they represent coarse dolomite rhombs with equant sub to euhedral crystals. And these are cloudy cores with clear rims and represent our inclusion-rich phases. And then the latest stage is the pore filling and fracture filling replacement dolomite, and they also have an enriched signature of dolomitizing fluid. And so you can see the paragenetic sequence that was developed from this. So moving on to our fluid inclusion thermometry. So fluid inclusions are fluid-filled vacuoles that are sealed within minerals that provide a direct means of examining fluids that were present in ancient diagenetic environments. So they represent an important constraint for eliminating some models, because from this we can get our temperature and our salinity of the fluids when the crystal formed or was dolomitized. So on the chart that you see here, I've placed several dolomitizing models and where I think they would go. So you can see that mixing zone dolomites would represent a lower temperature and lower salinity on your fluid inclusions. And then as you go up to evaporative and then burial dolomitizing models, then you increase the temperature, and for burial, you increase salinity as well. You increase both temperature and salinity. So this is the process for the fluid inclusion microscopy. So I start with a standard thin section. I analyze it under a microscope to identify fluid inclusion bubbles or vacuoles, and then I map out the assemblage of fluid inclusions and conduct an analysis. So microthermometry is carried out using a Lincoln heating and freezing stage, and then I observe specific phase changes within the inclusions. So the behavior upon heating and freezing is indicative of the fluid composition and the minimum trapping temperatures of the fluid when it formed. So for my results, this is a chart of the homogenization temperature for all of my dolomite phases. So you can see here clearly that there is a large assemblage of fluid inclusions that were trapped around 100 degrees C. And then you also have a later stage of fluid inclusions that were trapped above 200 degrees C. When I look at the homogenization temperatures of inclusions for individual dolomites, I can see that my cement dolomites have a wider range of entrapment for the fluid inclusions, but in general, the majority of them are also trapped at 100 degrees C. As I move to my D1, which represents the early stage dolomitization, I see that the window of entrapment for my homogenization temperatures are smaller. And then for D2, it represents the smallest range. So the least variability within my homogenization temperatures. When I look at the salinity of the fluid inclusions that I measured, most of them are very saline with a weight percent sodium chloride around 24. And then I also have a later, a less saline phase at 12 to 14 weight percent sodium chloride. So looking at the salinity, comparing the salinity for each individual host mineral, my D2 represents the least variable one, and that has a very clear salinity range of about 23 weight percent sodium chloride. D1 is very variable, and so that has salinity that ranges from about 12, with the majority of them occurring around 24 weight percent sodium chloride. And then my cement dolomite represents the most saline, with the average of that being around 24 weight percent sodium chloride. When I compare the salinity with the homogenization temperatures for each host mineral, I can begin to make interpretations about the dolomitizing environment for each of my phases. So putting this into a density ellipse, I can see that my D2 mineral, which represents the late stage replacement dolomite, is the most confined. So it represents the dolomite that has the most restricted environment. And then my D1, I can see a trend in that as well, where I am both increasing in temperature and salinity as I increase in depth. And then for my cement dolomite, that is the most variable, and there's no clear trend for that mineral phase. So in conclusion, I was able to look at each stage for my dolomites in the Smackover in Alabama. So I have an early stage fine-grained dolomicrite that has shallow burial and low temperature during early diagenesis. And so then I was also able to look at the later stage replacement dolomites, which are coarse equant subteuhedral dolomites with cloudy cores and clear rims that had really good inclusions in them. And I was able to get an average temperature of about 120 to 140 degrees with an average salinity of about 26 and ranging all the way up to 45. And then for my latest stage replacement, which fills the pores and fractures, that one shows the most enriched dolomite signature for the dolomitizing fluids with temperatures of ranging from 90 to 100 to 130 degrees Celsius with high salinity as well. So I'd just like to acknowledge my advisor, Dr. Marcello Minzoni, the Center for Sedimentary Basin Studies at the University of Alabama, the members of my committee, other people in my carbonate research group, Carmen Chauvet and Lucas, and then other people that have helped me conduct different analysis, Dr. Joe Lambert, who helped with the oxygen and carbon isotope, Dr. Rob Goldstein, who has helped guide me and teach me about fluid inclusions, and then other funding agencies. And so that concludes my talk. Thank you for your attention.
Thank you, Karina. So do you guys have any questions? Please just write it in the chat. So now that you have finished all the characterization, what exactly is going to be your next step? Is this your general model? Are you going to, how different is from the previous model that has been done in the same area?
Yeah, so like I mentioned, the previous models, there were many different ideas about the dolomitization in the area. So the next steps that I'm gonna do right now is to compare all the different previous models that and the data that they've collected with the fluid inclusion data that I have now, and see how those models fit in with the new data that I have. And then from there, I'm going to also look at another field within Alabama that is less dolomitized, so that I can get an idea of the starting conditions, and then have a better idea about the paragenesis and kind of the progression of dolomitization throughout Alabama.
Oh, wow. So you still have some, how far are you in your PhD?
So this is my 4th year of my PhD.
Okay. So it's still a little bit of more work to do. Any questions?
Hi, this is Jean. I have one question. So it's nice to see that you see different types of dolomite, and I understand what you're trying to do next with your analyses. Just, do you have any ideas of which one is critical and why it's happening where to control the best reservoir quality?
That's a good question. So one thing that I haven't done so far is look at petroleum fluid inclusions, which I think would be important for understanding the reservoir and which minerals are actually hosting the petroleum reservoir. So that's something that I want to do next, is to look under the UV light first to understand which dolomites have the petroleum in them, and then do an analysis on the petroleum fluid inclusions to get a better understanding of where those fit into the paragenetic sequence. Is that what you were asking?
Yeah, that's sort of exactly, because that's what's important to a lot of us who are working in the ministry. Right?
Yeah. So that is a next step that I'm going to take for this field specifically, is to first look at the under UV light for the petroleum ones.
Cool. Thank you. Good luck.
Yeah. Thank you.